Humans are an incredibly complex animal. Each of us has over 20,000 genes, tens of billions of neurons, and over 37 trillion cells. With that said, the question is, how did we get here? Let's explore how humans evolved on Earth, starting with the most basic form of life to exist. How did life on Earth begin? To answer this question, we need to start about 4 billion years ago, when the first thing to ever live was just beginning to form, and explain what the first form of life was made of, and how the ingredients that made it came together. The first component of the earliest life form is lipids. You're probably familiar with lipids as being fats called triglycerides or cholesterol, but the kinds of lipids that made up early life are a different subgroup called fatty acids. The really amazing thing about these lipids is that they are amphiphiles, meaning they are molecules made up of a hydrophilic or water-attracting head and a hydrophobic or water-repelling tail. What is so great about that? Well, it means that these lipids had the ability to naturally arrange themselves into sphere-shaped groupings of other lipids and form a protective casing called a lipid bilayer. Encased inside the lipid bilayer were amino acids and nucleotides, the molecules that make up RNA, DNA's single helix cousin. These spheres of lipids containing the ingredients for RNA are called protocells or protobionts, and were most likely formed in hydrothermal vents, fissures in the ocean floor that emit heated water and materials. Protocells are the first and most basic form of life on Earth, and we call one of them LUCA, or the last universal common ancestor, because every living thing is descended from it. So we know what LUCA was made of and how those ingredients came together, but that's not enough for it to be alive. It also needed to be able to self-replicate. Fortunately, the means to self-replicate already exist within the protocell. The trick is the RNA. You see, the nucleotides that make up RNA can arrange themselves in different patterns, creating differently shaped strands. Any of the 4 types of nucleotide in RNA can attach to any other nucleotide, but the secret to RNA's ability to self-replicate is that the bases of the nucleotides have only one type of nucleotide they can attach to. This is called base pairing. When chains of nucleotides base pair with each other and then split apart, that is how RNA self-replicates. Over the next billion years, random mutations within the nucleotides of replicated RNA would drive the increasing complexity of RNA chains through what is called chemical evolution. Strands of RNA that were more successful at resisting entropy, or more simply, better at self-replicating and not falling apart, survived and were able to spread their genetic information, and over the next billion years evolved the complexity required for unicellular life. Okay, that was a lot to take in, but now that we know what was the earliest form of life, we can explore how that life form evolved and branched to become the abundant biodiversity of species that we know today. The first branch on the tree of life happened 2 billion years ago, with the divide between bacteria and the branch that included archaea and eukaryota. The bacteria and archaea, which are both prokaryotic, are topics for different videos. For now, let's explore the lineages of the eukaryotes, the branch on the tree of life that contains plants, animals, slime molds, fungi, and protozoa. The domain eukaryota, or eukarya, is distinct from its ancestor LUCA, in that the organisms in this group's cells have a nucleus containing DNA within its lipid bilayer, instead of just RNA and proteins. There is some debate whether DNA first formed around the same time as RNA, or if it appeared around 500 million years later, but either way, organisms that self-replicate through DNA evolved after organisms that self-replicated through RNA. There are also other characteristics of eukaryotes that make them much more complex than their prokaryote ancestor, so many steps in chemical evolution must have occurred in those 2 billion years. There would be several branches occurring between 1.4 and 2 billion years ago when the eukaryotes diverged, branching into diphoda or the biconts, which became land plants and algae, and podiata, which includes things such as animals, protists, and fungi. Podiata then branched into crumbs, a clade containing protozoa such as Collodictyonidae and Mantamonadidae, and Uniconta, or Amorphia. 1.4 billion years ago, Uniconta split, separating the amoebozoa from its sister clade Obozoa. Then 1.3 billion years ago, Obozoa split to include Breviatia, Aposomonadia, and the clade Opisthoconta. The Opisthoconts belong to the clade of life that includes animals, fungi, and cholanoflagellates. Then 1 billion years ago, it split into Holomycota, which is the clade containing fungi, and Holozoa, which is the clade containing animals and their single-celled cousins. Phylozoa split from Holozoa 782 to 900 million years ago. Then Phylozoa split into Philisteria and Choanozoa. And then Choanozoa split into Choanoflagellata and Metazoa, or Animalia, meaning every life form in the next video will be an animal. In part 2, we will explore the lineage of the first animal, all the way up the tree of life to modern humans, and find out how we are related to some other animals along the way. There are quite a few more stages of life to cover in the next episode than in this one, but that should be easier to digest than the ancestors of plants, animals, and fungi, so don't feel overwhelmed if part 1 is a lot to take in. Be sure to check out part 2 to see how humans evolved, and subscribe so you don't miss out future videos on the evolution of different animals too. Until next time, see ya.